Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
John Carew Eccles 1957 monograph, The Physiology of Nerve Cells, represents a defining moment in 20th century neuroscience, a work of profound synthesis and experimental triumph, published by Johns Hopkins Press. This volume serves as the capstone for the body of research that would earn Eccles, alongside Alan Hodgkin and Andrew Huxley, the Nobel Prize in Physiology or Medicine in 1963. It constitutes a comprehensive statement on the functional mechanics of the synapse within the central nervous system, articulated at the very moment when the long-standing debate over electrical versus chemical transmission was reaching its denouement. The book is a monument to a particular mode of inquiry, the application of the intracellular microelectrode to the vertebrate central nervous system. This essay will undertake a direct and systematic evaluation of the book's internal claims, assessing on a section-by-section basis the evidence, arguments, and conceptual frameworks that Eccles presented to the world. We will explore the architecture of his reasoning, the power of his methodology, and the enduring structure of the physiological model he so masterfully constructed from the electrical whispers of individual neurons. Section 1. The experimental foundation, the spinal motor neuron, and the microelectrode. Eccles commences his exposition by establishing his experimental universe. The choice of the mammalian spinal motor neuron, specifically within the anesthetized cat, is a central and calculated feature of the entire research program. He presents the cell as an ideal preparation, valued for its large somatic size, which facilitates the difficult task of intracellular impalement, and for its well-defined, monosynaptic inputs from stretch receptors via the dorsal roots. This anatomical clarity allows for a clean experimental design where a known, synchronous volley of afferent impulses can be used to evoke a postsynaptic response in a specific population of target cells. The book's initial chapters are dedicated to validating this model system, detailing the surgical preparation and the revolutionary tool at its heart, the glass capillary microelectrode filled with a concentrated salt solution, such as potassium chloride. Eccles meticulously documents the properties of these electrodes and the criteria for a successful intracellular recording, namely a stable, high-value resting membrane potential of approximately -70 millivolts. The central claim here is methodological, that this technique provides a faithful, high-fidelity report of the electrical life of a single central neuron. The evaluation of this claim is overwhelmingly positive. The stability and magnitude of the recorded potentials provided irrefutable evidence that the principles of ionic gradients and membrane potentials, brilliantly elucidated by Hodgkin and Huxley in the squid giant axon, were fundamentally applicable to vertebrate central neurons. This act of experimental translation was itself a monumental contribution, bridging the conceptual gap between invertebrate preparations and the mammalian brain's complexity. The book's power flows directly from the confidence Eccles builds in his recording setup, presenting it as a reliable window into the soul of the neuron. Section 2. The excitatory postsynaptic potential as a depolarizing event proceeding from the stable baseline. Eccles directs his analysis to the consequences of excitatory synaptic action. He details the recording of a characteristic, transient depolarization following the stimulation of an excitatory pathway. This he terms the excitatory postsynaptic potential, or EPSP. His core argument is that the EPSP is a graded potential, its amplitude proportional to the number of activated afferent fibers, which serves to drive the membrane potential of the motor neuron towards the threshold for firing an action potential. The book's critical contribution in this section is the analysis of the ionic mechanism underlying the EPSP. Eccles presents evidence that the synaptic transmitter substance, upon binding to the postsynaptic membrane, creates a profound and indiscriminate increase in permeability to multiple ions. He deduces this from experiments attempting to find a reversal potential for the EPSP. By artificially shifting the motor neuron's membrane potential, He demonstrates that the ETSP amplitude diminishes as the membrane is depolarized, projecting towards a reversal potential near 0 mV. This value, sitting between the high positive equilibrium potential for sodium and the negative equilibrium potential for potassium, led him to the conclusion of a general ionic short circuit. The claim is one of functional generality. He posits a mechanism, less like the specific, voltage-gated sodium or potassium channels of the action potential and more like a temporary puncture of the postsynaptic membrane. In evaluating this claim from a modern perspective, Eccles' reasoning is both brilliant and a product of its time. His conclusion of a generalized permeability increase was fundamentally sound and represented the most logical inference from the available data. Subsequent research would, of course, refine this picture, revealing specific ligand-gated ion channels, like the AMPA and NMDA receptors, which are primarily permeable to cations, namely sodium and potassium, with calcium also playing a key role. The short circuit was an elegant and effective metaphor for a process whose molecular components were still entirely unknown. The book's claim, however, that excitatory transmission works by depolarizing the cell through increased ionic conductance remains a pillar of neuroscience. Section 3. The discovery and mechanism of the inhibitory postsynaptic potential. The intellectual climax of the physiology of nerve cells is undoubtedly its treatment of synaptic inhibition. Here, Eccles provides the first definitive, mechanistic account of postsynaptic inhibition in the central nervous system. This section directly confronts and resolves a long-standing mystery. He presents clear recordings of a transient hyperpolarization following stimulation of an inhibitory pathway, an event he names the inhibitory postsynaptic potential, or IPSP. The existence of this hyperpolarizing potential demonstrated conclusively that inhibition was an active process, a specific physiological event designed to move the membrane potential away from the firing threshold, thereby stabilizing the cell. This discovery itself was revolutionary, but Eccles' deeper contribution lay in his experimental dissection of its ionic basis. The book's most elegant piece of experimental logic is detailed here. By injecting current through the recording microelectrode, Eccles systematically varied the resting membrane potential of the motoneuron and observed the effect on the IPSP. He showed that as the membrane was artificially hyperpolarized towards negative 18 mV, the IPSP diminished in amplitude, vanished, and then inverted to become a depolarizing potential. This demonstrated a clear reversal potential for the inhibitory process. The value of this equilibrium potential for inhibition at approximately negative 80 mV was slightly more negative than the resting potential but far from the equilibrium potentials for sodium or calcium. This led Eccles to a stunning deduction. The inhibitory transmitter must be opening channels, selectively permeable to ions, whose equilibrium potential resides near this negative 80 mV level. The primary candidates, based on the Nernst equation which relates equilibrium potential to ionic concentrations, were potassium ions with an equilibrium potential around negative 90 mV, and chloride ions with an equilibrium potential near negative 70 mV. Eccles concludes that the IPSP is generated by a selective increase in the membrane's permeability to these two small ions. This claim was a masterpiece of physiological inference, explaining the biophysical nature of inhibition purely from electrical recordings. Later work identifying GABA and glycine as the principal inhibitory transmitters and their receptors as ligand-gated chloride channels confirmed the core of Eccles' hypothesis, cementing the IPSP characterization as one of the great discoveries in modern biology. The book presents this finding with the quiet confidence of irrefutable proof, settling decades of speculative theory. Section 4. Neuronal integration and the genesis of the action potential. Having established the elemental units of synaptic communication, the IPSP and the IPS, Eccles dedicates a significant portion of the book to their interaction. He advances a model of the neuron as a computational device, an integrator that performs algebraic summation of incoming signals. The central claim is that the neuron's firing output is determined by the continuous, moment-to-moment arithmetic of depolarizing and hyperpolarizing influences. He provides compelling evidence for both temporal summation, where repeated IPs from a single source build upon one another, and spatial sonation, where simultaneous EPs from different sources combine their effects. The motoneuron's vast dendritic tree and soma are presented as the stage for this summation. Furthermore, Eccles advances a crucial argument regarding the site of action potential initiation. He posits that while the dendrites and soma are the sites of graded potential integration, they possess a relatively high threshold for firing. The action potential, the allo-nothing signal, is instead generated at a specialized, low-threshold region, the axon hillock or the initial segment of the axon. The summed potential from the soma and dendrites, he argues, spreads electronically to this trigger zone, and only when the depolarization at this specific location reaches its lower threshold does the neuron commit to firing a spike. This concept of a separation between an integrative zone and a trigger zone was a profound conceptual leap. It provided a coherent framework for understanding how a cell could process a multitude of graded, analog inputs and convert them into a single, digital, all-or-nothing output. The evaluation of these claims finds them to be remarkably durable. The principles of spatial and temporal summation are taught as fundamental tenets of neurophysiology today. The identification of the axon initial segment as the primary site of spike initiation due to its high density of voltage-gated sodium channels remains the canonical model, while contemporary research has unveiled a far more complex picture of active dendritic processing and backpropagating action potentials. Eccles' formulation provided the essential, first-order model of neuronal computation that has structured the field's thinking for over half a century. Section 5 Presynaptic mechanisms and the unity of transmission. In the latter parts of his monograph, Eccles turns his attention to the presynaptic side of the synapse and to broader principles of neuronal organization. He marshals the evidence for the chemical nature of transmission, highlighting the irreducible synaptic delay's brief but consistent lag between the arrival of a presynaptic action potential and the onset of the postsynaptic potential. He argues this delay is incompatible with direct electrical continuity and is best explained by the time required for transmitter release, diffusion across the synaptic cleft, and binding to postsynaptic receptors. This argument, while building on work from the neuromuscular junction by Katz and others, provides strong confirmation within the central nervous system. Within this context, Eccles becomes a forceful advocate for what is now known as Dale's principle. He formulates the principle as the law that a single neuron makes use of the same transmitter substance at all of its synaptic terminals. If a neuron was found to be excitatory at one synapse, all its other synapses would likewise be excitatory. If its collaterals were shown to mediate inhibition, as with the Venture cell interneuron, then its primary axon terminal would also be inhibitory. This claim embodies a principle of metabolic and functional uniformity for each neuron. In assessing this claim, one recognizes its immense heuristic power. For decades, Dale's principle was a foundational assumption in circuit analysis, providing a crucial simplifying constraint for deciphering complex neural networks. The idea brought an elegant order to the daunting chemical complexity of the brain. The subsequent discovery of neurotransmitter co-transmission, where a single neuron can release multiple distinct signaling molecules, has shown that the biological reality is more intricate. Yet, as a working hypothesis based on the evidence of 1957, Dale's principle as presented by Eccles was a productive and logical generalization. It reflects the book's overall project to find unifying, powerful physiological laws that govern the seemingly chaotic operations of the nervous system. In conclusion, the physiology of nerve cells stands as a masterwork of scientific argument and exposition. Its profound and lasting impact derives from the direct and rigorous evaluation of its own central claims, all grounded in the unassailable evidence of the microelectrode. Eccles' methodical progression from establishing a reliable experimental system to defining the fundamental particles of synaptic communication (EPSP) and the IPSP, and then assembling them into a coherent model of neuronal integration, provides a template for scientific reasoning. The book's supreme achievement was the definitive biophysical characterization of central synaptic inhibition, transforming it from a theoretical necessity into a tangible, measurable physiological process with a plausible ionic mechanism. The claims advanced within its pages, from the nature of postsynaptic potentials to the principles of neuronal summation and the logic of the axon initial segment, have become so foundational that they form the very grammar of modern neuroscience, while subsequent discoveries have added layers of molecular and computational complexity. The fundamental physiological architecture that Eccles described in 1957 remains remarkably intact. The work did more than simply present findings, it constructed a durable conceptual framework for understanding how nerve cells communicate, a framework built not on speculation, but on the precise and eloquent language of bioelectricity.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.